Hello students, in this video lecture we are going to discuss about the blood cells. First we are going to discuss about the RBC, red blood cells. These RBC are also called the erythrocytes. The main RBC, mainly RBCs are biconcave, circular and non-nucleated. At the time of origin, nucleus is present, but eventually that is ejaculate during the maturation process. Biconcave shape of RBC increase the surface area that is helpful to carry the oxygen and other nutrient substance. The RBC having the plasma membrane that is called Doman's membrane, it is highly permeable and maintain the inflow and outflow of ions such as chlorine, HCO3 and also that phenomena is known as Doman's phenomena. Due to the presence of spongy your RBC does not having the endoplasmic reticulum, so that the inner skeleton of RBC is made up by the cytoskeleton, that is spongy cytoskeleton. In the RBC, higher cell organelles like mitochondria, Golgi complex is absent. Due to the absence of mitochondria, anaerobic respiration takes place in RBC. In RBC, enzyme of glycolysis process are present while enzyme of Krebs cycle are absent. In RBC, carbonic enzyme is present which increase the rate and formation and dissociation of carbonic acid by the 5000 times fastest. Okay, in RBC, red colored represent pigment hemoglobin. In each 26.5 Cr molecules of Hb are present. Molecular weight of each molecule of hemoglobin is 76,200. In composition of RBC, 60% of water, 40% soil part is present. Only Hb consists 36% of total weight of RBC. Now we are discuss about the hemoglobin. Hemoglobin is a component that is formed by the two components. It is composed of two components, heme that forms the lipid part that is near about 5% and globin that is protein part that consists 95%. Heme, first we are discussing about the heme, that is iron, and iron present in the form of Fe+2, while in the muscles myoglobin is present where iron is present in the form of ferrous. Each molecule of Hp carries 4 molecules of oxygen, and another part of the hemoglobin is globin, that is the protein part that composed of 4 polypeptide chains. Polypeptide chains are 4 types. These are alpha polypeptide chain, beta polypeptide chains, gamma polypeptide chain and delta polypeptide chain. Oxygen binding capacity of hemoglobin is more in fetus than adult. Size of RBC is near about 7.5 mu. Changes of RBC size, that is called aniocytosis. Due to the vitamin 12 deficiency, RBC become larger in size, that is called macrocytosis. These are immature RBC which are destroyed in the spleen. In these RBC, amount of hemoglobin is normal. Due to ferrous deficiency, RBC becomes smaller in size, that is called microcytes. They are also destroyed in the spleen. In these RBCs, amount of hemoglobin is less. shape of RBC, as you know that in the RBC is biconcave due to the ejaculation of nucleus. RBC change the shape during the transportation uh, by the blood vessels, that is called polycaliocytosis. Uremia RBC becomes irregular in shape, that may cause the anemia. Sickle cell anemia RBC become sickled shape. If RBC is kept in hypotonic solution, it will shrunk. In hypotonic solution, it must burst. Lifespan of RBC in human is near about 120 days. In human male, RBC count is 15.5 plus minus 2.5 gram per dl, and human females it is 4.5 millions, and newborn babies 6.8 millions. Increase of RBC count, that condition is called polycythemia. This condition occurs at hilly stations. Decrease in RBC count condition is called anemia. These are so many different types. And formation of RBC process is called erythropoiesis organ, which is produced RBCs as erythropoietic organ. Hormones which stimulate erythropoiesis is called erythropoietin, synthesized by kidney. First RBC is produced by yolk cell. During the embryonic life, RBC are produced by liver, spleen, placenta, thymus gland. In adult stage, the RBC is produced by red bone marrow, which filled in tubercles of spongy bone. 1% of RBCs are destroyed daily, but the same number of RBC are formed in the blood. Destruction of RBC occurs in spleen, so spleen is graveyard of RBCs. Spleen stores excess blood corpuscles, so it is called blood bank of body. In resting and slow flowing blood, the RBC follow pele called Rulex by adhering together due to the surface tension. Fibrinogen favors relaxing condition. Minute bite of destruction red blood cells is known as hemoconia. The ghost of RBC is made up of its plasma membrane. The formation of RBC is, as you know, that is called the erythropoiesis. Erythropoiesis, in which this you can see in this image, the formation occur in the red bone marrow. First, the red bone marrow differentiate into the myeloid tissues. The myeloid tissues differentiate into the myeloid cells, which are called the stem cells. These stem cells are modified in pre-erythroblast cells. Due to the mitosis, the pre-erythroblast cells are changed into the erythroblast, which there is nucleus is present, cell organelles are present. The erythroblast change into the normoblast due to the maturation by the vitamin B12 and folic acid. The size and shape as RBC. Here the nucleus is absent and cell organelles are absent. When normocytes enters into the blood, it is called erythrocytes. So it is from all about the RBCs. In next video lecture we are discussing about the